Thank you, Eleanor. Okay, so hi everyone, welcome to this ESID Junior Journal Club. I am Selket Delafontaine. I'm a PhD student of the Laboratory of Inborners of Immunity at the KU Leuven in Belgium. And I am very happy to welcome you all, and I'm very honored to introduce you to our speaker of today, which is Zhiyong Liu. And he majored in pharmaceutical engineering during his undergrad studies and received a PhD in immunology in China. And during his doctoral research, he investigated the molecular mechanisms governing antiviral innate immunity using mice models. And then to study antiviral innate immunity in Natura, he joined the Casanova lab at the Rockefeller University as a postdoctoral associate in 2019. And his current work is mainly focused on discovering new genetic determinants of childhood herpes simplex virus encephalitis and also post-herpes simplex virus encephalitis autoimmune encephalitis. So today he will introduce his latest work on a novel etiology underlying childhood herpes simplex virus encephalitis. And his presentation is entitled Encephalitis and Poor Neuronal Death Mediated Control of Herpes Simplex Virus in Human Inherited RIPK3 Deficiency. So it's going to be super interesting. Don't hesitate to ask any questions. You can put them in the chat box or save them for after the presentation, which is going to take about half an hour, and then we have 10 minutes of discussion. And finally, certainly remember to tune into the next session, which is on the 19th of October. Voila. So let's enjoy Zhiyong's presentation now.
Great. Thanks. Let me share my screen first. And yeah. And I hope you can see my full screen now, right?
Yes, we can see it very well.
Okay, awesome, great. Hi everyone. First of all, I would like to thank Selket for the nice introduction, and thanks to all the ESID organizers for giving me this great opportunity, and I really appreciate it. And it's also a great honor for me to open the new season ESID Journal Club this year. And this work is a single patient study, but we are not alone along the way. And we have GG's group from NIH to help us to do the the iPS cells from the patient's skin fibroblast, and we also collaborate with the Stewart Lab from the Sloan Kettering Cancer Center to make the neuron differentiation from the iPS cells. And also we also have Edward Mokaski group. They are the expert for the virology and also the cell death. So we got a lot of support from them for the biochemical assays. And also the patients were recruited from France. So we also got a lot of support from the physicians from France and elsewhere in Europe. So this is really a team effort, and we could not make it without them. And the single patient study, as you know, has been always challenging, especially when you are trying to show a novel genetic etiology of a disease. However, as John Casanova always said, it should be encouraged because it can be the first step in understanding of a group of human disease, and thereby revealing a critical pathways underlying the physiological and the pathological processes. And actually, this work was presented in the last year's ESID meeting in Sweden. And one of my colleagues helped me to take a photo for my posters. And I can still feel alone for my poster. You know, I was not there, and I cannot help him to answer the questions there. You know, "Wish You Were Here" is an old song from a rock band named Pink Floyd from English. But today I will not talk about the rock music. But you know, the virus we are going to talk about today might really rock some people's brains. So firstly, let me introduce some background of our studies. So human herpes virus is a group of enveloped double-stranded DNA virus, and from the outside to in, it contains the envelope and the tegument and capsid and the core. And the core contains the genome of the virus. Overall, there were around 8 human herpes virus, and 6 of them was reported to could invade the central nervous system of the human beings. But today I will mainly focus on a virus named oops, could not do something. But today I will mainly focus on a virus named the herpes simplex type 1 virus. Short name is HSV1. Actually, HSV1 is a common virus in the world, and most of the people were infected. And the primary infection usually occurs in the childhood. And after the primary infection from the epithelial cells, the virus could transmit into the peripheral neuron system, and named the trigeminal ganglions. And there the virus could be latency, but the virus could be reacted periodically. And most of the circumstance, the clinical phenotype is where it's benign, and sometimes people will suffer the cold sores around the mouth and like that. But there is a very rare cases the virus could also infect the central nervous system and lead to a very severe disease named herpes simplex encephalitis. Short name is HSE, which is a very rare disease with occurrence around 2 to 4 cases per million people a year in the world. Even rare, this disease is also the most common sporadic viral encephalitis in the Western countries, and perhaps in the whole world. Without treatment, it is typically fatal disease, and most of the patient could benefit from the antiviral drug named acyclovir. And after with the treatment with the antiviral treatment, the mortality could join to around 20%. But most of the survivors still suffer from a very severe neurological sequelae. So as you can see on the right panel, the brain image from MRR, the affected people displayed a very specific vulnerable to the HSV1 infection in the central nervous system, and it showed the inflammation and swelling in the brain tissue. And from the history, in early 1941, HSV has been identified as a causal agent for HSE. But for a long time, the pathogenesis of this disease remained unexplained. So with the high preference of the HSV1 infection in the world and the very low prevalence of HSE led to the search for a human monogenic basis for this disease. So in the last, oops, okay, got some trouble for the slide. In the last decades, our lab and others have found that the inborn errors of immunity, especially for the central nervous system specific cell intrinsic immunity, could underline the etiology of HSE. And the published disease-causing gene for the HSE mainly focus on the toll-like receptor 3 and the type 1 interferon pathways. what we found so far could only explain around 5 to 7% of the patient in our cohort. So we hypothesized that other inborn error immunities could also underline the etiology of childhood HSE. So today I will introduce a patient from France. She suffered 2 episodes of HSE at 6 months and 17 months of age. She could benefit from the antiviral drug acyclovir, and unfortunately, she also suffered the post-HSE autoimmune encephalitis after that. So here the left upper panel showed the first episode of the brain image. You can see the lesion in the left temporal hemisphere. So, oops, let me show. Yeah, so we do the whole exome sequencing from the patient and also the parents, and we finally a PhD student, a previous PhD student from the lab at Vado, find the patient harbors component heterozygous variant in a gene named RIPK3. So far, the patient parents are healthy. So what is RIPK3? RIPK3 is a key molecular kinase involved in a programmed cell death named necropsis. RIPK3 could interact with RIPK1 and then MLKL to make a complex named necropsome. And then the fully activated MLKL will translocate it to the cell membrane and induce the cell rupture and the following inflammations. So RIPK3 dependent necropsis has been fully studied in the downstream of toll-like receptor. So except for the toll-like receptor pathways, RIPK3 dependent necropsis could also involved in the downstream of other receptors, such as the interferon receptor, toll-like receptor 3, toll-like receptor 4 receptor, and also another nucleotide a receptor named DAI. So RIPK3 contains the N-terminal kinase domain and the C-terminal rim motif. So except for the necropsis, RIPK3 could also interact with RIPK1 by his C-terminal rim motif to mediate the apoptosis. So RIPK3 mediated apoptosis is RIPK1 dependent. So except for the cell death, RIPK3 could also mediate the cell death independent manners by mediating the pathway such as NF-κB, MAP kinase pathway, autophagy, and inflammation. So come back to the 2 variant of from the patient for RIPK3. As you can see, the stop-gain variant loses the C-terminal rim motif, which is predicted to be loss of function for RIPK3 dependent cell death. However, overall, the frame shift variant looks fine, but still lose a small part of the C-terminal of the protein, which might be had a mild effect for the RIPK3 effect. And this variant is a big challenge overall for the project. So how about the mice study for the RIPK3? So it has been reported by several groups that the RIPK3 knockout mice were highly susceptible to various kind of viral infections, such as HSV1, influenza virus, Western Nile, and Zika virus. So overall, this studies could divide it mechanically into 2 parts. One is the cell death dependent manners, and another is cell death independent manners. So our working hypothesis is that the autosomal recessive RIPK3 deficiency results in HSE due to the deficiency in the cell death dependent and or cell death independent antiviral immunity. So what we did firstly is to test the expression of the two variant RIPK3 in the HT29 RIPK3 knockout cell lines. Why we choose this kind of cell line is HT29 cell line is a human cancer colon cancer cell line. Why we choose this cell line is because this kind of cell express high level of RIPK1 and RIPK3 and it's a very good cell model to study the RIPK1 and RIPK3 mediated cell death. So overall, at the MR level, the two variants showed a similar expression level of, with the wild-type RIPK3. And as expected, at the protein level, the two variants lead to the truncated form of the proteins. And what surprised us is that the frameshift variant protein level is much lower than the wild-type and the stop-gain variant of RIPK3. So one hypothesis is that the frameshift variant RIPK3 protein might be unstable and prone to the excessive protein degradation. So to test this hypothesis, we did the transient transfection in the HEC293 T cells for the RIPK1, and then treat the cell with the protein synthesis inhibitors named the CHX, and then we test the RIPK1 protein half-life times. So as you can see on the left panel, the half-life time of the frameshift variant is much shorter than the wild-type and the stop-gain variant, which means the frameshift variant RIPK3 protein would be prone to the protein degradation. And to go further for the mechanism, we found that RIPK3 protein could be protein degradation could be mediated by the proteasome pathway and the lysosome pathways. So we tried these pathways inhibitors, and finally we found only the proteasome inhibitors could rescue the frameshift variant protein in the HT29 cell lines. And then next, we started the function of the two variant in the RIPK1 HT29 overexpression system in terms of the RIPK3 dependent cell death in the downstream of toll-like receptor 3 and the TNF receptor. So here the poly-IC is the ligand for the toll-like receptor 3, and TNF is the ligand for TNF receptor. BV6 is the inhibitor for the cell survival. So here the combination PB or TB here could induce the cell to apoptosis. And BV6 ZVAD is the inhibitor for the apoptosis. So the combination here, PBZ or TBZ, could induce the cell to necropsis. So as you can see from this assay, the stop-gain variant is complete loss of function for toll-like receptor 3 or TNF-mediated apoptosis or necropsis. However, the frameshift variant looks to be hypomorphic in these assays. To go further, for the virology part, so we also started the DEI-mediated necropsis after the HSV1 infection, because DEI is a kind of ISG and the basal level is very low. So what we did is we overexpressed the DEI in the HT29 cells, and then after that, with the after the stable transduction for the DEI, we infected the cells with HSV1. So overall, what we found is that cell with the stop-gain variant RIPK3, these cells are resistant to DEI-mediated cell death after the HSV1 infection. However, at the same time, HSV1-induced cell death is impaired in the frameshift variant expressed HT29 cells. So far, what we found here is that the stop-gain variant of RIPK1 in the patient is completely loss of function for RIPK3 mediated cell death. And the frameshift variant had a mild phenotype, which is hypomorphic for the RIPK3 mediated cell death. in both the toll-like receptor 3, TNF receptor, or DI mediated cell death. So next, we transferred our study from the molecular level to the cellular level. So firstly, we tested the RIPK3 expression, the MI level with the patient cells. Overall, the MI level is in the range of the healthy controls. And then when we going deeper to test the proportion of the two variants for the RIPK3 transcript in the patient cells, what we found is that most of the around 80% of the RIPK3 transcript in the patient cells contains the frame shift variant. However, there's only 20% of the RIPK3 transcript contains the stop-gain variant, which indicate that the stop-gain variant might variant transcript might undergo the nonsense mediated MI decay. And then how about the protein level? What surprised us is that with the Western blot, we could not detect any RIPK3 protein by Western blot in the patient cells. And then we used the protein degradation inhibitors to treat the cell, and just found all the protein degradation inhibitors could somehow rescue the RIPK3 expression in the patient cells. Next, we also tested the patient's fibroblast for the response of toll-like receptor 3 or TNF mediated cell death. And what we found is that the patient cells are resistant to the both apoptotic or necroptotic cell death and in the patient cells. And this phenotype could be rescued by overexpressed the wild-type RIPK3 in the patient cells. Our previous study has found that the type 1 interferon, type 1 interferon immunity would be important for the antiviral, for the anti-HSV1 infection. So we also tested the interferon responses with the patient fibroblast. Overall, we found the patient cells could have a normal response or the induction for the type 1 or type 3 interferons. However, we found impaired induction for the some inflammatory cytokines such as IL-6, IL-8, and CCL3. So we also used the bulk RNA-seq with the patient fibroblast to test the patient cells the patient cells cellular response to the HSV1 infection at the genome-wide levels. So overall, in brief, what we found is that the patient fibroblast had a normal cellular response to the HSV1 infection at the genome-wide transcript levels. So our previous study have found that the deficiency of the anti-HSV1 cell intrinsic immunity, especially for the cortical neurons, could underline the pathology of HSE. So the patient's dermal fibroblasts were reprogrammed into the iPS cells, and then we diff the iPS cells into the cortical neurons for the viral assays for HSV1 infection. So what we found here with the iPS-derived cortical neuron is that the patient cortical neuron, iPS-derived cortical neurons were highly susceptible to the HSV1 infection comparing to the healthy control. And this phenotype could be rescued by the beta interferon pretreatment. And more importantly, we found the patient iPS-derived cortical neurons was highly resistant to HSV1 infection-induced cell death. And to illustrate the causality between the genotype of the RIPK3 deficiency and the clinical phenotype of the patient, so we also started the RIPK3 in the isogenic model in the iPS cell lines. So we used the CRISPR-Cas9 system to knock out RIPK3 and then differentiated into the cortical neurons. So consistent to the patient iPS-derived cortical neurons, the RIPK3 complete knockout cortical neurons are more susceptible to the HSV-1 infection, and the phenotype could also be rescued by the beta interferon pretreatment. And we also found the RIPK3 knockout iPS-derived cortical neurons are resistant to HSV-1 infection-induced cell death comparing to the healthy controls. So in summary, what we found here is that in the healthy cortical neurons, the patient the cells could defend the viral infection, the HSV-1 infection by the toll-like receptor 3 and a DI-dependent apoptosis, necropsis, and the inflammation cytokines induction. However, in the RIPK3 deficient cortical neurons, the necropsis is completely abolished, and the apoptosis and the inflammatory cytokines induction would severe impaired, which result in the out of control of the viral replication for the cells, and then could explain the pathology of the clinical phenotype of the HSE patient. So this work is supported by the Jonathan Casanova, my supervisor, and Dr. Shenying Zhang. And firstly, I would like to say thanks to the patient and her family. They helped us a lot. And also, I would like to thanks to our HSE group and also other cell members from the New York City and Paris. And more importantly, I would like to say thanks to our collaborators, especially for the Stewart Lab. They helped us a lot for the iPS cortical neuron differentiation. And his previous postdoc Oliver, now he's the PI in Italy. And also I would like to thanks Louis Gigi's lab from NIH. They helps us for the iPS induction from the patient fibroblast. And also I would like to thanks Greg Smith's lab from the Northwestern Universities for the support of the virology part from the ESID, and also thanks the Edward Mukaski lab and for his a lot of comments and suggestions from our biochemical assays for RIPK3. They are the real expert from this field. And his previous postdoc, now he a PI from the Louis State University named Hongyan Guo. They are still the collaborators with us for the following studies. So, and also thanks for the fundings for us. And that's all my presentation. And I would like to answer any questions. Thank you all.